Hello, everyone. My name is Brittany. Today we're going to be talking about COVID-19. COVID-19 is a serious global infectious disease which has recently had an outbreak globally with nearly 191 million cases and around 4.9 million deaths worldwide. It is part of a family of viruses called coronaviruses that infect both animals and people. This particular one originated in China at the end of 2019 in the city of Wuhan, which has about 11 million residents. COVID-19 can cause symptoms very similar to that of the flu, which can include the fever, dry cough, which are the most common symptoms, fatigue, muscle, body aches, nasal congestion, loss of smell and taste. Those are the most common symptoms. Not everyone has all of those symptoms, but most people will have some of them. And then some people are also asymptomatic, meaning they don't have any symptoms. Those are the scary ones. Severe cases can also lead to serious respiratory diseases like pneumonia, which can also lead to death. Those that are most at risk are elderly or people with underlying medical issues such as heart problems or diabetes. So since the original outbreak, scientists have been able to create a vaccine for COVID-19, but right now they're struggling because there's been a new variant of the Delta variant of COVID-19, which is causing people who have already become immune to the coronavirus to contract that new variant of it. So right now they're working on potentially getting a booster shot for that. Some viruses, such as the coronavirus, which also causes COVID-19, only contain RNA, which means that they rely on infiltrating healthy cells to multiply and survive. Once they're inside the cell, the virus uses its own genetic code, that RNA in the case of COVID-19, to control and reprogram the cells, turning them into virus-making factories. In order for a virus like COVID-19 virus to be detected early in the body, we have to use a test for RT-PCR. Scientists do this by converting the RNA to DNA in the process called reverse transcription. They do this because DNA can only be copied or amplified, which is the key part in the real-time RT-PCR process for detecting the viruses. So scientists amplify a specific part of the transcribed viral DNA hundreds and thousands at a time. Amplification is so important that instead of trying to spot a small amount of the virus among the millions of strands of genetic information, scientists have a large enough quantity of the target sections of the viral DNA to accurately confirm the virus is present. So how this all starts is a sample is collected from parts of the body where the COVID-19 virus gathers, which is mainly the back of the throat or the nose. Most often, a nasal swab will be inserted into the very back, up into the nasal canals. The sample is then treated with several different chemical solutions that remove the substances such as proteins and fats that extract only the RNA, so that the only thing extracted is the RNA present on the sample. The extracted RNA is a mix of the person's own genetic material, and if that's present, the virus's RNA. The RNA is reverse transcribed to DNA using a specific enzyme. Scientists then add the additional short fragments of DNA that are complementary to specific parts of the transcribed viral DNA in the virus. If the virus is present in the sample, these fragments attach themselves to target sections of the viral DNA. Some of the added genetic fragments are used for building DNA strands during amplification, while others are used for building the DNA and adding marker labels to the strands. which are then used to detect the virus. The mixture is then placed into the RT-PCR machine, and the machine cycles through temperatures that heat and cool the mixtures to trigger specific chemical reactions that create new and identical copies of the targeted sections of that viral DNA. The cycle is repeated over and over to continue copying that target section of the viral DNA, and then each cycle doubles the previous number. 2 copies become 4, 4 copies become 8, and so on and so forth. And then the standard real-time RT-PCR setup usually goes about 35 cycles, which means that by the end of the process, there's about 35 billion new copies of sections of the viral DNA created from each strand of the virus present in the sample. So as new copies of the viral DNA sections are built, the marker labels attach to the DNA strands, and then the release of the fluorescent dye, which is measured by the machine's computer, are presented in real time on the screen. The computer tracks the amount of fluorescence in the sample each cycle, and when a certain level of fluorescence is surpassed, that confirms that the virus is present. Scientists also monitor how many cycles it takes to reach this level in order to estimate the severity of the infection. The fewer the cycles, the more severe the viral infection is. The RT-PCR is also used to detect past infections, which is important for understanding the development and the spread of the virus, as the virus is only present in the body for a specific amount of time. Other methods are necessary to detect, track, and study past infections, especially for those of us who, like I had mentioned before, are asymptomatic and don't actually have any symptoms that we show. That's all for today.